Hello, biologists. We were working on translation in part 1 of the video. And so hopefully you have translated successfully codons 1, 2, 3, and 4 using the codon wheel built into gizmos. And now we're going to bring in our tRNA, our transfer RNAs, which act like little shopping carts going around and picking out the amino acids needed to line up with the codons here. So let's look at how the tRNA are working here. Let's look closely. The tRNA have an anticodon that matches up with the codon. So here's the codon on the RNA, and here's the anticodon, the one that fits, that matches up in the tRNA. And this anticodon is attached to a tRNA that will only bring the correct amino acid here. So this UAC anticodon will bring methionine, which is the code, the mRNA code AUG means methionine. And there it is, there's methionine. So that's the first one. That's usually the first amino acid in every protein. And then as the next tRNA comes in, the next codon is GCA. Using what you've learned about base pairing, select the tRNA with the anticodon that would pair to GCA. So we know that C goes with G, and that G goes with C, and that A goes with U in RNA. So that would be C, G, U. So let's see if we can find CGU. There it is. And now we have 2 amino acids, and they're going to make a peptide bond. In our model here of making a protein out of different amino acids, a peptide bond is the little click you see in these kind of click beads. And here, it's going to be a chemical bond. So there's a peptide bond between these 2 amino acids that helps link all the amino acids together and make a chain of amino acids that will eventually fold into the right conformation and make a protein that works in the body it's being made in. The process continues to translate the mRNA codons into an amino acid chain. So tRNAs keep bringing in different amino acids depending on what the code is. So maybe the next code codes for this amino acid. This is going to come in and get attached to the rest of the growing protein chain. Let's continue translation here. All those tRNAs come in really fast, and even these are all colored blue. You can think of them as different colors because they're different amino acids. There's 20 different amino acids. This process continues to translate mRNA codons into an amino acid chain. That amino acid chain is going to be the protein, until it gets to a stop codon. A stop codon says, don't bring any more amino acids, we're done. And you can see the kind of the stop sign here that is the stop codon. And so the protein is leaving the ribosome here. Translation stops when a stop codon is reached. Stop codons are either UAA, UGA, or UAG. So there's, just like there's several different codes for some amino acids, there's different codes for stop. So when it comes time for the stop codon, then a release factor, which basically pushes the protein out and says, you're done, pushes it out of the ribosome, gets it out into the cytoplasm. The amino acid string then will fold into a protein, and the protein will be ready to do its job if and only if all those amino acids are in the correct order and the correct amino acids have been put into the protein. If there's been a mutation and the codons are messed up, then you can't have a working protein. Of course, there could be a silent mutation, and we'll learn what that is, but if you have coded for a different amino acid, you may not have a working protein. So we can review how amino acids shape proteins. They have to be in the right order. You have to have the right amino acids, and it has to be the right length. DNA transcription is making RNA from DNA, and codons and anticodons. Codons are on the RNA transcript, and they tell the tRNA which tRNA to come next and what amino acid should be next in the chain. So now let's go back to the case and see if we can help Lucy here. We've gone through Lucy's lab data. Changes in the order of nucleotides are often called mutations. A mutation in a gene will give a different order of nucleotides in the mRNA. That means that the order of amino acids in proteins will change and the protein may not work correctly. In other words, the beads in the protein chain here won't line up correctly, which means it won't fold correctly, which means it won't work. Lose your shape, lose your job. Let's look at 3 DNA mutations that could cause Lucy to have ADA-SCID. An active site mutation. And the active site is a very specifically shaped part of the protein that grabs onto this toxin and breaks it apart. If you don't have a working active site, which is shown here, you're not going to break down this toxin. Here it's successfully broken down, here it's still whole, and it's going to impact white blood cells. Stop codons end the translation of a protein. If a mutation creates a stop codon early in the amino acid sequence, so if the amino acid's humming along and instead of doing the whole amino acid, it hits a stop codon early and stops, it's going to be missing part of the protein if it stops too early. The protein will not be, if it stops and it's too short, if it's too early, it won't fold correctly and it won't Here's a mutated sequence with early stop codon, doesn't work well. The enzyme doesn't work well and doesn't break down deoxyadenosine. There could be a tRNA error. If a mutation caused tRNA molecules to carry the wrong amino acids, proteins would not be made correctly. But that would probably affect all of the proteins because that messed up tRNA would work with the ribosome to translate all of the proteins in the body of a person who has messed up tRNA. So it's supposed to carry methionine, but for some reason it could be messed up and be carrying arginine. That would really mess up all the proteins in the body. So right now it's time for us to pick a hypothesis. We can look at, let's look at Lucy's gene length. She has 1,500 base pairs. That's right. She has 1,500, her mRNA is correct. It has 1,500 nucleic acids, which is normal. But her amino acid sequence, she's supposed to have 500 amino acids, and she only has 283. It's likely that the amino acid chain was humming along just fine, and then it hit a stop codon and it stopped too early. It didn't do the rest of the job and create this part of the protein. It stopped too early and made a shorter protein. That's why it only has 283 amino acids instead of 500. It has just over half the amount of amino acids it's supposed to. So that's a really good clue that tells us that it's probably a stop codon mutation. And you can pick stop codon and you can describe why you think the stop codon mutation is correct. In the meantime, let's go look at our lab sheet. So we did the first one together, methionine, and you now have the information to describe what happens in transcription with words like ribosome, codon, anticodon, amino acid, and peptide bond. Oh, let's change this to translation. What is unusual about Lucy's ADA protein production? Use the word amino acid in your answer. So this right here, when we look at a normal ADA protein would have 500 amino acids, but Lucy only has 283. So effectively, you've kind of cut the protein in half. Something in the way the RNA is working with the tRNA, something in translation isn't quite right. And you hit an early stop codon and you only get about half the protein. That can't form a correct working enzyme and it will not work. What happens if there's an active site mutation in ADA? The shape is going to be wrong. It's not going to fold up into a working enzyme. What does a stop codon mutation do? If we put a stop codon in a place where it's not supposed to be, if it's too early, we're only going to get half our protein. We'll only get half the number of amino acids brought in by tRNA because that mRNA will have a stop in it before the end of bringing in all those amino acids, and we'll get a shorter protein. And that's not going to work for Lucy. She needs a working full-length protein to break down a deoxyadenosine and be healthy. How would a tRNA error alter the translation of proteins? We talked about this a little bit. There's an anticodon on the tRNA that matches the codon on the mRNA. And it would bring in the incorrect amino acid. And if it brings in incorrect amino acids, if every codon that's supposed to be methionine is now arginine, you're going to have a lot of miscoded amino acids in your protein, and the protein's not going to fold correctly. And that would be all the proteins and all the places where methionine is supposed to be. So probably her body wouldn't have any working proteins if she had a tRNA error. So choose the mutation that you think is causing Lucy's ADA enzyme not to work, form a hypothesis, and explain your answer using data from Lucy's lab work. So if you choose one of these, which you need to, explain from Lucy's lab work why you think what you do. Make sure you have a hypothesis, it's the stop codon mutation, and you have a reason, you have something to back up your hypothesis. Now, let's go do some experiments. I'm just going to type some garbage in here. This is not the correct answer. You guys are going to have to come up with the correct answer on your own. Let's do some tRNA analysis and try to figure out what the correct answer certainly is. Let's investigate if the translate of Lucy's ADA enzyme. We'll need to find out if Lucy's tRNAs are carrying the correct amino acid. So there's a AUG mRNA codon, there's a UAC anticodon, and that should be methionine here. To build proteins correctly, a tRNA must carry the correct amino acid for its particular codon. So if this is not methionine, your body's going to have a problem building proteins. Here are some of the tRNA molecules in Lucy's white blood cells. Let's see if the tRNA number 1 is carrying the correct amino acids for its anticodon. So here's the anticodon, CCA. We know the base pairing rules are A goes with U and G goes with C. So this is GG, and then U goes with A. Let's submit that. All right, let's use the codon wheel to decode GGU. So we click G, we click G, we click U, and we know it's glycine. And it's carrying glycine. Bonus. That's exactly what we need. It's supposed to have glycine. It does have glycine, so we're good. She's good on that tRNA. Let's figure out the rest of the tRNA. So there's a whole bunch of tRNA. There's lots of different codes, and it's just scanned all of them and said all the tRNA are normal. Obviously, it would take a lot of time to do this, but it's unlikely that there would be any working proteins if you had a tRNA error in all of your tRNA. Maybe a few of them could have a mutation, but if you had a mutation in all of your tRNA, you wouldn't be making any functional proteins, and you probably wouldn't be alive. So if you guess tRNA error, you might want to change your hypothesis. If you didn't guess tRNA error and you guessed something else, you might want to explain. You're going to want to explain how this data supports your hypothesis. So don't just say I'm right. You can say the tRNA is not the problem, so it's probably still whatever I guessed, whatever I hypothesized. So let's investigate mRNA and see if there are any mutations. The sequence of nucleotides in Lucy's ADA mRNA transcript could have been changed by a mutation. The mutation could cause the ADA enzyme not to work. Let's scan for mutations here. Obviously, this would take more time and a lot of lab work, but through the magic of computer simulations, we can find out this very quickly. 2 mutations have been found in Lucy's mRNA transcript. And a lot of people have mutations, and some of them don't do anything. We found a mutation in ADA's active site. This codon has changed from GAC to GAU. Select the codon to investigate. It's supposed to be aspartic acid. Now let's check. Lucy is GAU, it's different, it's mutated. Oh, but it still codes, because there's 2 codes for aspartic acid. It still codes for the same thing. So the first mutation did not change the amino acid. A mutation that does not change the amino acid sequence is known as silent mutation. There are multiple codes for most of the amino acids, and so if it just has a code that's different but it codes for the same amino acid, that's called silent, because it doesn't actually change the protein. It will change the mRNA, but because there's several codes for each amino acid here, alanine has 4, valine has 4, glycine has 4, if it just changes to another code for the same amino acid, then it's silent and it doesn't really matter. Let's go to the next mutation. So it's supposed to be arginine, but what does Lucy have? Lucy has UGA. UGA. Ooh, that's stop. Ouch. So she's putting a stop codon in the middle of her mRNA where it is not supposed to be. That could cause big problems. So explain how this stop codon and the silent mutation in the active site both support your hypothesis. So we know that there's a stop codon in the wrong place. That's a mutation. We know there's a mutation in the active site, but it's a silent mutation. So you should be discussing the silent mutation and the stop codon mutation. And how they support or refute your hypothesis. Our hypothesis is correct. You've shown that Lucy's tRNA are carrying the correct amino acids. There was a mutation in the active site, but it's a silent mutation and did not change the shape of the active site. You found a stop codon mutation at codon 284. This has shortened Lucy's ADA enzyme from 363 amino acids to 283 amino acids. This means that her ADA enzyme does not work. So we figured out what's going on. Let's move forward. So mutated enzyme isn't all there, doesn't work. Let's see what we can do to help Lucy. Lucy's immune system needs white blood cells with working ADA enzymes. This will help her white blood cells break down the toxin. So we're going to take stem cells out of her blood, and we're stem cells that can become white blood cells are found in your bone marrow. Lucy's will have an operation to move a sample of her bone marrow, probably from her pelvis. Most bone marrow donations, if one person donates bone marrow to another, are taken from your pelvis. In the laboratory, the stem cells from the bone marrow are placed in a petri dish under conditions that keep the cells healthy. A technology called CRISPR-Cas9 has been used to replace the incorrect DNA sequence of the ADA gene with the correct DNA sequence. And CRISPR is pretty cool. It hasn't been used much on humans, but it's a new technology that's emerging that has a lot of promise to do a lot of things. It can actually go in and snip out very carefully just parts of DNA and put new DNA in there to replace the old stuff. The stem cells, the repaired stem cells, are allowed to duplicate and grow, so there are millions, and then they can put them back in Lucy. The stem cells then change into white blood cells, have working ADA enzymes, and these white blood cells will be able to break down the toxin and survive. They'll strengthen Lucy's immune system, and because they're in her bone marrow, they'll stick around in her bone marrow and keep producing white blood cells that have the correct ADA gene in them. So let's go look at our lab sheet here. So explain how the tRNA scans that all the tRNA had correct amino acids support or does not support your hypothesis. Write a new hypothesis if you need to. Explain what a silent mutation is. Remember, there was a silent mutation in the active site. It had a different codon, but that codon happened to code for the same amino acid, so it didn't change the shape of the protein. Remember, each amino acid has several different codons that code for it. Does the mutation scan support or not support your hypothesis? write a new hypothesis if necessary. If you guessed there was a problem with the tRNA, then you need to write a new hypothesis and explain how the discovery of the stop codon in the middle of her mRNA, where it's not supposed to be, supports or does not support your hypothesis. Here's just a little recap on the stem cell therapy. And we're heading into 28, 29, and 30. Almost done. So let's make a prediction how the data will change after treatment. Lucy has below normal white blood cells, so we're hoping those will come up. Her HIV proteins are not present, and we don't think they're going to pop up. She has, she's supposed to not have more than 13 nanomoles per liter of deoxyadenosine, and she has 34. So we're hoping that comes down to protect her white blood cells, because that's a toxin. Deoxyadenosine is a toxin to her white blood cells. Let's analyze Lucy's blood here. Wow, her white cells came way up. Yay, our treatment worked, at least to create more white cells. HIV proteins still aren't present. Her deoxyadenosine is now right in the normal, so it went down. Apparently I didn't make the correct predictions. Oh, that went down. Oh, that went up. Oh, what is it now? Okay, they're saying what is it now? I thought it was like how it changed. So she is right in the middle, right in the middle, and right in the middle. So she's normal on everything. I didn't read the question very well. So our predictions were correct. Her white blood cell went up, her HIV protein stayed the same, and her deoxyadenosine went down. So explain how you found that Lucy had ADA-SCID. Well, she had high deoxyadenosine. That's the toxin that was killing her white blood cells. She didn't have HIV proteins in her blood, so we knew that she probably had ADA-SCID because of that deoxyadenosine. I'll spell that out for you. Deoxyadenosine. There you go. That is not the correct answer. You need to provide more details. How did the stop codon mutation stop her ADA protein from working? Well, remember, her protein was supposed to be about this long, but because of the stop codon in the middle of the RNA, the ribosome read the stop and just went right in the middle of making the protein, and she only got about half of what she needed, a little bit more than half of what she needed of her protein. So it didn't fold correctly and it didn't work. The scan of Lucy's ADA gene showed a mutation in the active site, but it was silent. It coded for the same amino acid, so it didn't change the protein, and so it wasn't a problem. How did stem cell therapy make her better? They took out her cells, took out the bad DNA, put in good DNA, put the cells back in her bone marrow, and they will continue to make the correct version of white blood cells for the rest of her life, because they actually changed her DNA. Now, not many therapies can change someone's DNA. This is very rare, but we hope for young people with genetic disorders, this can become more common. Okay, the last question is writing wrap-up. Use the words transcription, translation, DNA, mRNA, tRNA, ribosome, order, amino acids, and protein folding in your paragraph. I will not give you the answer, but I will give you some tips to get your answer going. Remember, transcription takes RNA from DNA. Translation uses RNA to make protein. Nucleotides are part of RNA and DNA, but they're different. mRNA is transcribed, leaves the nucleus, and goes to the cytoplasm to work with the ribosomes. Nucleus. The ribosome puts the amino acids in the correct order so the protein can fold correctly. That's a pretty good beginning. You kind of know some of the major concepts that you need to include. That is not the answer. Don't use that as your paragraph, but you can certainly build on those ideas. Thanks so much for watching. This is Dr. B signing off on the Gizmos protein synthesis video.